discussion about ways to maintain and care for your mental health during grad school, and particularly the challenging times during COVID during grad school, which makes everything crazier. To the graduate students out there, my message to you is just that your faculty know the stress you're going through, we care about the stress you're going through, and we want to help you. I know that grad school can be such a trying time. For me personally, I know when I was getting ready to defend my dissertation, I was fighting suicidal thoughts pretty much every day of my life. I've been faced with some kind of mental health challenge, and my personal message to you is that if you're going through anything and I can help connect you to resources or just be a ear to listen, please reach out to me and let me do that. It would mean the world to me. Faculty, I also want to mention that you are one of the biggest influences of how your graduate students' mental health is, that their healthiness comes from you. And so if you would attend this event, it would mean more than I could possibly describe here, just to see a face of a committee member or their own PI in this meeting with Dr. Guest. So if at all possible, please try and make the event. So, all right, that's my personal plug, just something I'm really passionate about that I wanted to take a few seconds to talk about. Here's the overview of my talk. I'm going to introduce you to the people who have done the work, both the members of my lab and also the collaborators that we've been lucky enough to work with. I'm going to brag about their accomplishments and how this has all led into the papers that we put out as a group and the grants that we've gotten as a group. Then I'm going to give you a general background, starting with the field, and kind of narrow that down into how my group fits into the field. And ultimately, I'm going to give you the choice of how the talk proceeds. I've prepared two different talks. My lab works on HPV viruses in the genital tract, as well as HPV viruses in the skin. And based off of the introduction, and I'm going to let you pick which story you want to hear more of. You'll do that through the chat when that option becomes available. But let's get going. If you're part of the Acker-Chalmers complex, you've probably seen my group. Lorena mentioned our team building activities. They're there just to break down artificial barriers of communication and really to build camaraderie in the lab. And I think it's worked. We're a tight-knit group who really all appreciate each other. There we go. Over the time I've been at K-State, I've had 16 undergraduate researchers in the lab. I currently have 3 students. 2 of my former students are off in grad school, which is awesome. And you can see the accomplishments of these folks in the lab: 16 scholarship presentations, not just here at K-State, but they've gone other places, including to the Capitol in Kansas, to give presentations. They have authorships, 4 more pending, in fact. So they're doing wonderful. Here are the lab's alums, at least all the ones that I could find pictures of. And here's my 3 current students, Zachary, Taylor, and Elizabeth. And I really think that this current crop of students might be the best yet. They all have authorships already, even though Elizabeth's the only senior. Zachary's is technically pending, but I think it's coming any day now. And Taylor had her first authorship basically as a freshman. So I am immensely proud of you, and you're doing wonderful. I also have had research assistants or technicians in the lab. I use this as a gateway to grad school. My goal is to get people who want to leave the lab as soon as possible and get into grad school. And we've been successful so far. 4 out of 4 of our research assistants have entered grad school. Dalton's here at K-State. He's actually in my lab. Monica went to the UK to Bristol and already has her PhD and is postdocing. Jasmine just started at the University of Washington / Fred Hutchinson Cancer Center, and Vaibhav is at Ohio State University. As technicians, this group is presenting at national conferences. They're authoring papers. Really did a wonderful job, and they all earned their admittance into grad school. Now, grad school. Man, grad students are the absolute best, right? They come in, they're so full of energy, and they kind of recharge you. And I've gotten to work with 7 of them and watch how they've progressed from, that early state of excitement up to like a little bit more disgruntled later on, but super productive. I've had 3 more permanent members of the lab. Dalton and Chung Quinn are PhD students, kind of getting close to finishing their degrees. Stephen got a master's degree in the lab and graduated a couple years ago. And I've also had 4 rotating students, which have been cool and added variety to the lab. We had Emily, Candy, Laura, and Peter all rotate through the lab. They've got plenty of authorships, and this group is speaking not just at national conferences, but also at international conferences. I've gotten to mentor 2 people with their doctorates. Andrew Holcomb had his MD and Sebastian his PhD. Sebastian's still a postdoc in the lab and doing wonderful things with us. And Andrew's currently a surgeon at Nebraska Methodist Health Center. These fellows have won awards, they've won fellowships, they're authors on papers, and again are presenting at conferences. So everybody's doing great. And I have to say that, traditional in K-State biology tenure talks is that you brag a little bit about yourself. And my biggest brag is that I've been able to be friends with all of these people because they really have enriched my life, and I appreciate all of you. We've had collaborators too. Collaborators are wonderful. They expand what you're able to do by adding expertise into your field that you don't have before. I just want to mention people who we either have papers out with or we have papers pending, or they're coming up in the near future at least. And that would be Phil, Katsura, Nigel, Kevin, Geelong, Linda, Rachel, Lisa, Osama, Sufi, and Sen. Oh, sorry, I should say that I also included people I've gotten a grant with. Okay, so speaking of grants, we have had some grant success, as Lorena mentioned early on. So far, my group's raised $1.5 million in direct support, and that's directly fed into our research program, which has been wonderful. We've also helped to enrich the research environment at Kansas State University, helping to bring in $12.4 million. You can see the breakdown of that by years at K-State going into the future. And the sources of funding have come from non-traditional sources like direct philanthropic support from the Les Clow family, as well as local support from our cancer center. And we have a grant from the Department of Defense, and we're supported by a couple of grants from the National Institute of Health right now. We've been productive. We've put out papers. 2020 has particularly been a good year. So we have 12 peer-reviewed manuscripts overall. In 2020, we have one paper that's pending at PLOS Pathogens. It says, I looked it up right before this. It says basically the decision's been made and they're just waiting to email me, which makes me super anxious. But that aside, we had a high-profile review and 4 primary research authorships papers this year. So we're doing good things. I'm really proud of it. But that's a bunch of generic, right? So it's, what are these papers on? What do we actually research? And to get into that, I want to start with the question that the field is really interested in, which is, how do human papillomaviruses cause cancer? In other words, how can an infectious disease lead to a tumor? And the hope isn't just to answer that question because we're all curious, but rather to answer that question so that we can do something about it. And, I'm not some like ridiculously deluded person. I don't think anybody's going to take our paper and change treatment, but you hope that you build incrementally forward so that the next generation can do even more than you were able to do. We got a pretty broad audience here, so I want to make sure we all have the same background going in. And I think it's best at this point to break down the virus I'm particularly interested in into its roots. Human papillomavirus comes from 3 roots. The first is human. That one doesn't really need any definition, but just to say that this virus infects humans. Papilloma is the second root, and it's a little bit less approachable of a word, but essentially it means warts, and HPV, or human papillomaviruses, do cause warts. It's uncontrolled growth of cells. Now, finally, you've got virus. And if you're like me, you thought you knew what a virus was in middle school. I thought it was that thing that you got when it was cold out generally and it made you miss a day of school or something and feel a little sick. Since middle school, I keep learning more about viruses to the point that today I'm convinced that A, I don't really know what a good definition is exactly for a virus, and B, that whatever definition you come up with, there's going to be a virus that's found that doesn't exactly fit it. For today, we're going to call them obligate intercellular parasites. I think that works generally well. Obligate meaning they're dependent on living intercellularly or inside of our cells. And parasite meaning that that relationship really is all take and no give. It's Dolly Parton. Okay, so that's a long introduction. We haven't gotten to much details, and you've got to be asking yourself at this point, how much more time is he going to waste on just general background and random bits of information? But what we just learned, that HPV lives inside of our cell, tells us something very important. It tells us that HPV is very small. And that's critical for a couple of reasons. The first is that it facilitated a blatant act of vandalism that nobody here has noticed. If you remember a few years ago, Jesse talked to us in his promotion talk about Big Blue Stem, this wonderful prairie plant that's about 1 to 2.5 meters tall with this like it's completely amazing root structure that goes way down, and he put up this banner showing up his wonderful organism of interest on the wall in the atrium. And the first time I saw it, I thought, that's really cool. And then I started to get more and more jealous that HPV wasn't up there. So one day in the middle of the night, I came in and I hung out over this balcony and I drew a life-size representation of HPV on the other side, thinking that, all right, when my coworkers come back in on Monday, they're going to see the diversity of life. It goes from this tiny, tiny thing to this great big plant. But unfortunately, or maybe fortunately because I didn't get arrested, HPV is tiny. It's 55 nanometers in diameter. Now, obviously that's a joke. I didn't do this, but it is important because HPV's small size means it has limited capacity to pack DNA into that capsid. HPV isn't just small because it's a virus, it's considered small among viruses. This means it doesn't have the luxury of having multiple genes to do the same function, and it's simplistic in that way, but that creates a type of complexity wherein HPV has to be multitasking with its genes. And this really plays a lot of role in what HPV does and the stories I'm going to tell you today. I want to go one more metaphor to compare this genetic complexity in maybe a little bit more tangible terms. If we compare the genetic complexity of the cells that HPVs are infecting, so the human genome, each cell of our body has 25,000 genes. We can represent each of those genes as a penny to have an idea of how massively complex our cells are. So here's a stack of 50 pennies. Before they had those machines in the grocery store where you just dump your change in and it counts it for you, we used to count 50 pennies and roll them. This makes me sound like a dinosaur, but here are a roll of those 50 pennies and a group of 20 of them. So that's 1,000 pennies right there. To be the complexity genetically of every cell in your body, you'd have to have 25 times that amount in this 25,000 pennies that you see on your screen right now. So in comparison, here's HPV's genetic complexity. And I'm being generous here. Plenty of HPVs just have 7 genes. So to put it another way, HPV, or the human genome in pennies, is complex enough to pay for you and a loved one to go to a very nice meal. In fact, in Manhattan, Kansas, I think you might struggle to spend $250 on a meal for 2 people at a restaurant. In contrast, HPV's genetic complexity is the equivalent of leaving an incredibly insulting tip at any restaurant you might go to. This matters a lot for biology, right? Because HPV needs to manipulate the cells that it infects in order to survive. 8 genes, 7 genes, it's not enough to make all the proteins and all the enzymes that you need to be a productive virus. So HPV manipulates the cells into giving it what it needs. And it does this in a lot of ways, but the way that my lab focuses on is HPV's manipulation of the cell's response to damaged DNA. Essentially, HPV has invested a lot of its energy into co-opting to manipulating the cell's response to damage repair such that the cellular environment is productive for HPV's replication. I'm going to get a little less metaphoric, a little less abstract, and show you an actual picture of HPV's genome at this point. I want to point out that this genome is actually circular. I'm showing it linearized here out of the review that came out from our lab this year, just because it makes it a little easier to to talk about. You'll notice that the genes are organized into 3 colors. We've got red genes, green genes, and yellow genes. Those red genes, E6 and E7, they manipulate the host cell environment. They're the ones that are responsible for convincing the cell to care more about replicating HPV than it does about its own genome's integrity. E1, E2, and E5, these are responsible for replicating the viral genome. And I have to pause here because I know some HPV folks are in the audience and I am super oversimplifying everything by describing these 3 genes this way, so please forgive me. Finally, the viral capsid, that shell, that 55 nanometer shell, is made from the L1 and the L2 proteins. Now, I personally am a bit of a reductionist by nature. I think that we have to understand the details of what's going on before we can understand the broader picture of life. And so my group focuses really just on the E6 and the E7 genes from HPV. You could accuse us of primarily focusing on E6 even. But we're interested in these primarily because they're the ones that do the bulk of the manipulation of the cellular environment to change things. Now, at this point, you might be getting a little bit annoyed at the generic use of the term manipulate, right? What does it really mean? Is it trying to get HPV to, or the cell to change its mind about something? No, of course not, but what exactly do I mean? And the reason that I've been abstract and vague with that term is that the answer really depends on where the HPV infection occurs. Kind of like us, what we need out of our environment depends on where we live. I know that the coat I needed in winters in New Orleans is not the coat that I need in winters in Kansas. So HPV family is a very large and very diverse. We tend to think of, in pop culture at least, HPV as being a singular entity, but really it's a very large, diverse family of viruses. I'm going to click to a more simplified version of this so that you more clearly see the 5 genera that HPV is divided among: the alpha, the beta, gamma, mu, and nu papillomaviruses. And infections with these viruses cause clinical manifestations ranging from rather benign warts all the way up to incredibly deadly cancers. My group is focused in on the 2 genera of HPV that are most closely connected to cancers in humans. We know that the high-risk members of the alpha papillomavirus genus cause cancers in the head and the neck and throughout the anogenital tract. In fact, they handed out the Nobel Prize before I started my postdoc for that discovery. The members of the beta papillomaviruses are more tenuously connected to cancer and may cause cancer in some settings, seem to cause cancer in certain immune-compromised settings, in fact. So these are the 2 groups of HPVs that we tend to work on in my lab. I'm going to go in a little bit more detail at this point to tell you about the 2 of them and kind of answer that question of what type of manipulation was I talking about when I said HPV manipulates its cellular environment. So those beta genus HPV infections, they're occurring in our skin, right? And so the thing about your skin is that it's frequently exposed to UV. When the cells in your skin, those that HPV might be infecting, get exposed to UV, they want to stop growing so that they can repair the UV damage. This puts them at odds with HPV because the beta HPV infections, to be productive, they need proliferating cells. And so beta HPVs are invested heavily in getting cells to grow despite UV damage. You can see how this would lead folks to hypothesize that HPV infections in the skin, convincing your skin cells to ignore that they were damaged, might connect to skin cancer, because we know well that the repair of UV, or at least the UV sunburns, is connected to skin cancer. And like I said, there is some evidence of this happening in certain immunocompromised individuals, genetic disorder, as well as after organ transplants, where you see increased beta HPV infections associated with an increased prevalence of skin cancers. On the other side of this coin, the majority of adults have actually been infected with beta HPVs. Thing is that they tend to be transient infections and not around at the time you get a tumor. So this has led to a debate in the field actually of how dangerous, how mutagenic, how oncogenic beta HPV viruses are, and we're hoping that our work contributes to that discussion. Alpha HPVs are a completely different story, right? So they occur in the genital tract and in the throat, at the base of your tongue or in your tonsils. These are areas without a lot of sunlight. I mean, you have to imagine some pretty absurd scenarios to get sunlight in the back of your throat, like maybe you fall asleep at the beach backwards snoring or something. But most of the time, alpha HPVs aren't having to fight the cellular response to UV damage. And as a result, their relationship with DNA damage is actually very different from what the betas do. So alpha papillomaviruses actually cause DNA damage. And then they wait for the cell to come and try and repair that damage, and yoink, they steal those repair factors and use them to replicate their own genomes. This leads to a lot of cancers. In fact, alpha papillomaviruses cause essentially every cervical cancer out there. And cervical cancers kill someone every 90 seconds to 2 minutes, somewhere in that range. The statistics absolutely horrible. Worse of it is that these diseases and these deaths are occurring in impoverished nations, as you can see by the map here showing the incidence of cancers, cervical cancers, as well as deaths from cervical cancer in the world. Now, anytime you give a talk about HPV and you're trying to convince people to pay attention past the introduction, you deal with this question. Isn't there a vaccine against HPV? Isn't this problem already solved for us? And the short answer is no, I don't think so. Here's why. HPV infections, they don't instantly cause cancer. It's not like you get an HPV infection today and tomorrow you have a cancer. Instead, most of these infections take much, much longer. 10 to 30 years is a range of when they cause cancer. It can be earlier than that, but the point is that it takes a long time for an infection to cause the cancer. Gardasil, that very first HPV vaccine that got approved by the FDA in the US, wasn't licensed until 2006. So even if on the day it was licensed, everybody in the US got vaccinated, really quick math tells you that we would only now be seeing decreasing rates of cervical cancers. We'd be seeing cervical cancers still for a long time to come. So it's worth studying them to see if we can improve things. But not only that, I have to tell you that unfortunately, there is no such thing as a vaccine fairy. I can certify this 100%. It's not even on Google image searches. I had to come up with this creation on my own, where you can pretty clearly see I've pasted a needle and a syringe over a traditional fairy. This is the closest we have to a vaccine fairy. In reality, the US is facing tides of regrettable anti-vax movements and vaccine hesitancies that have Gardasil and other HPV vaccination rates tragically low. Okay, here's the part where you get to interact. Remember these books from like the 90s? I love them. You read a couple of pages into them, it tells you a bit of a story like we just did here, and then it lets you pick how that story continues. Now, almost all the time with these books, you end up dying. I promise nobody's going to die here based off of their choice. Okay, here are your two options. I could tell you more about how beta HPVs, particularly the E6 protein from these beta HPVs, induces genome instability, and we could spend the rest of this talk contemplating to ourselves whether that might increase the risk of cancer. Or I could tell you the story of how alpha papillomavirus oncogenes cause DNA damage by blocking replication stress responses. And we could instead have a conversation about how this makes cervical cancers responsive to existing therapies and how it lets us predict ways that they might gain resistance to those therapies. So what I want you to do is to go into the chat function on your Zoom and just to type alpha or beta to let us know how you want this story to progress. And we're gonna tally it up live as we're going, and we'll move forward based off of that. So Dalton's gonna be doing that. And meanwhile, I'll give you some ways to choose. So for the betas, here are the folks that worked on it. You can see their beautiful faces. Here's the folks working on alphas. Man, good luck tallying all of those, Dalton. All right, here are the papers that have come out. We've got basically 5 papers on either side with a couple of other things that might be shared between them. And ultimately, what I'm asking you to do, I didn't tell my lab I was going to do this. You get to decide who in my lab gets bragging rights for the tenure talk. Do you want to hear the beta HPV story or the alpha HPV story? Completely your choice. I'm going to let this run for about another 30 seconds or so to let Dalton tally everything up. Please get your vote in. And like I said, don't be shy if you're not a K-State person. We want everybody who came to the talk to make the choice with us. Cool. I'm so happy everybody's participating. Thank you. Sam. All right. Got about another 10 seconds to get in your answers before we're going to have a final tally and have to move on. All right. Dalton, whenever you've got a tally, let me know and I'll move forward with the talk. You can just post it in the chat, please. And I didn't realize how hard this might be to tally. All right. Dalton says betas have won, so we're going to move forward with the beta HPV story. Thanks, everybody. Here we go. Let's hear about Team Beta, who has just won bragging rights. I'm going to tell you about how beta HPV6 induces genome instability, or how it obstructs DNA damage repair, and other types of mechanisms meant to keep your genome safe. Now, while I'm doing this, I ask that you spend some time contemplating whether you're okay with the risk that these viruses might be causing, because non-melanoma skin cancers are not a particularly deadly cancer, but they are very costly. So we spend about $5 billion as Americans each year treating these cancers, and there's over 5 million new non-melanoma skin cancers diagnosed each year. And you know, like we all probably knew long before we came into this talk, these cancers are associated with UV damage, sunburns, and things that cause your skin ill health. Now, when I started my postdoc, and from that time on, I've been interested in this question of whether beta HPV infections might increase the risk of non-melanoma skin cancers. And at at the base of it, this seems like kind of a crazy idea. How could a virus increase your risk of cancer? But in fact, infectious agents cause about 15% of all cancers, so it's not that insane of a question. Oh, man, before all of you voted for beta HPVs, I probably should have told you that this option came with a pop quiz. You don't have to actually put your answer out there publicly. You can just think about it to yourself. Here, it's a one-question quiz. The question is, genus papillomavirus, genus beta HPVs are blank residents of the human skin. You have 4 options. It's multiple choice. Are they helpful, inconsequential, not there, or harmful? And, you know, honestly, there's only one answer that's truly wrong for this. So let's walk through this. Is there any evidence, if you picked A, is there any scientific evidence that you're right, that genus beta HPVs are helpful? Well, yes, there is. And in fact, a high-profile paper came out last year suggesting just this. They took mice, the ones on the left side of your screen were infected with a mouse version of beta HPV. The ones on the right were not infected with the virus. They exposed all of these mice to the same amount of UV, and the ones that didn't get infected with beta HPVs, or the mouse equivalent at least, were the ones that got skin cancer. And so this study went on to suggest that the role of beta HPVs is to prime the immune system, helping protect us against skin cancer. So if you said that, there's definitely a part of my field that would agree with you. What about if you said inconsequential? I think my postdoc mentor falls most squarely into this category. And the reason for that is that there's no evidence that beta HPVs are expressed in tumors. And that is the hallmark of an oncogenic virus, traditionally at least. So what actually happens when beta HPV infections are studied is that the most of their expression peaks before you have a cancer. And by the time you have a non-melanoma skin cancer, the viral load is so low, it's less than 1 copy per cell. It's basically just not there. And so people have concluded with scientific rationale and basis that beta HPVs are rather inconsequential residents of the human skin. What about not? Now, this was the only one that we can actually answer definitively with scientific evidence is absolutely not correct. Beta HPV infections are incredibly prevalent. We pass them apparently around among family units and with loved ones. And in fact, by the time we're adults, the majority of us are seropositive for beta HPVs. Now, seropositivity means 3 things. It means you got infected, you had an immune response, and then that we were able to detect it. So this means you really, really, really were infected. So sorry to say this, and I hope nobody voting for my tenure is going to hold this against me, but if you answered not, you just boarded the fail train, you are in fact wrong. So what about harmful? This is where the majority of the talk is going to center around the evidence that beta HPVs might be harmful residents of the skin. Now, we said that they weren't persisting in the tumors, so how in the world is a virus that's not even around when there's a tumor going to cause a cancer? Like, how's it going to cause that cancer? And there are prominent members of my field that suggest that a hit and run mechanism is the way this works. Here's how that would work. So you would have a beta HPV infection long before you got the skin cancer. It would coincide with a sunburn or a heavy UV exposure, and it would enhance the mutagenic potential of that sunburn or UV exposure, which is a fancy way to say that it would make the sunburn more likely to cause cancer. And then those mutations on their own, independent of the virus, are capable of continuing to drive tumorigenesis. The truth is that right now, the field, although maybe more people are leaning toward that they're not causing cancer, isn't currently sure about the answer to this question. So it was kind of a tricky pop quiz. There's only one answer that we have definitive scientific evidence for. The others are more open. And so my hope, my career goal is that by retirement, hopefully, I will have contributed to a field that has together helped answer this pop quiz from my tenure talk. Before you can tell anybody about, this is funny, there's like a chat going on right now from my lab debating who actually won the counting for which story we heard. Anyway, that aside, the E6 from these beta HPVs, if you're going to talk about E6 and HPV, there's a percentage of your audience that is going to associate E6 with P53 degradation. And I just have to tell you at this point that beta HPV E6 proteins do not degrade P53 by and large. Instead, they manipulate the cellular environment by interacting with the histone acetyltransferase P300. P300 is a very big protein. It's intrinsically unstable except for when it's phosphorylated in its CH3 domain by a cellular kinase AKT. This stabilizes the protein. And what Heather Howey showed nearly a decade ago was that some beta HPVs, HPV5 and 86, which I'll use as representative HPVs moving forward, bind that CH3 domain and block phosphorylation of P300. And this destabilizes the histone acetyltransferase. You can see the immunoblot showing this here, where HPV5 and 86, beta HPV6s that bind P300, cause less P300 in the cells. A HPV E6 that binds P300 more loosely does not, nor does HPV 16E6, which binds P300 in a different location. So these are the two proteins I'm going to be telling you about. Their way of manipulating the cell is that they reduce P300 levels in the cell. So we wanted to see what genes this was impacting, and I can show you the data here. This is what genes have their expression changed by a reduction in P300, and this really indicates P300's role as a master transcription regulator. In this volcano blot, anything that is blue, every blue dot is a singular gene that has had its expression significantly changed by a reduction in P300. Now, on the left side of the y-axis are genes that had reduced expression, and on the right side are genes that had their expression increased. Now, you can ask the question of what do these genes have in common, what cellular processes are they enriched for, and when you do that, you get a pretty good idea of what might have motivated a virus to evolve the capability of reducing the amount of P300 in cells. Namely, there's a lot of immune processes that are dependent on or influenced by P300. As a postdoc, I found that that wasn't the end of the story, that actually P300 promoted the expression of DNA repair genes, namely ATM, ATR, BRCA1, and BRCA2. We had a tool for testing the P300 dependence, namely we delete the P300 binding domain of beta HPV6 to give us this delta E6 mutant. And you can see in the purple bars that the mutant is unable to decrease ATM, ATR, BRCA1, or BRCA2 expression. But I want you to notice that there was some protein left in all of these. Somewhere around 20, 30% of all of these repair factors were still there. And that led to the question that I've been trying to ask or answer at K-State of, is what's left actually still capable of facilitating a repair response? So here, we focused really centrally on ATM and ATR for a lot of this work because ATM and ATR direct or orchestrate the repair of UV damage. Now, they activate themselves in an autophosphorylation event. So ATM is going to phosphorylate itself, ATR will phosphorylate itself. And throughout this talk, phosphorylation is going to be an indication that a repair gene or protein has become activated. Now, this leads to downstream signaling. For ATM activation can cause double-stranded break repair, cell cycle arrest. ATR can also cause cell cycle arrest. It can promote the repair of UV damage. And if neither of these happen, you'll get a replication stress cell, which ATR can also promote the cell's resolution of replication stress. So Jasmine Snow in the lab went about asking how 86 beta HPV affected the activation of ATR. You can see our vector control cells response here on the left are LXSN cells, where after UV, phospho-ATR levels rise up to the point so that the cell can take care of that DNA damage. 86 expression attenuated or reduced this response. There's still some activated ATR, but not nearly to the level you're seeing on the left side of your screen. She then asked what downstream consequences were resulting from ATR activation being reduced, and she found that check 1, the protein that should induce a cell cycle arrest, was also less phosphorylated when cells had 86 following UV. Finally, 86 blocked the phosphorylation and activation of an essential repair protein, XPA. Now, together, if you're not going to stop growing and you're not going to repair the damage, what's going to happen is your replication forks are going to eventually encounter a UV-induced lesion that they can't bypass. This is going to stress your cells, and they'll need to induce a translesion synthesis response. The indicator of that response that I'm showing you here is the formation of these polymerase ADA repair complexes. We can stain and view these using immunofluorescent microscopy, and you can see that in control cells, these little green dots build up over time, and any replication fork that's experiencing stress is going to be able to bypass it and tolerate that stress, avoiding the replication fork collapsing into a double-stranded break. But Jasmine showed that when you have 86 beta HPV expressed in those cells, this response doesn't happen. And that told us that, and we knew that as a result, beta HPV6 made it more likely that those replication forks collapse. I'm showing the data here to prove this point where we're using H2AX foci as a marker of collapsed replication forks, and our 86 that represented beta HPV6 doubles the likelihood of this happening. So you're getting more double-stranded breaks. Beta HPV has blocked at this point 3 cellular responses that should occur in response to UV damage. Those double-stranded breaks should elicit an ATM activation response. ATM should get activated. It does in normal cells. You can see the phosphorylation of ATM increasing in this vector control cells, but 86 is attenuating that response, especially if you look at the 6-hour and the 4-hour time points. So ATM is supposed to orchestrate repair of double-stranded breaks. It does this by activating a repair pathway known as the homologous recombination pathway, and that's going to repair double-stranded breaks. This is the first time that I'm using one of Jeremy Stark's assays, where you get basically GFP expression when a cell fixes a break by homologous recombination. And using this assay, we were able to show that our beta HPVs, HPV5 and 86, half the efficiency of homologous recombination in cells. Now, at this point, it struck me that the beta HPV story was almost like that cartoon villain who is continually foiling the cell's efforts to repair things. It's kind of on and on. And so I wrote this grant and I thought, you know what's definitely going to happen is that the cells are going to become more dependent on the backup system. When homologous recombination fails, you get an increase in non-homologous end joining. I felt really good about myself. I thought I had an answer. And we turned to another Jeremy Stark's cool tools to answer this question. So here's how this assay works. It allows us to measure the efficiency of this non-homologous end joining pathway, just that backup pathway for double-stranded break repair. You use Cas9 endonucleases to create targeted breaks in the endogenous genome. So no reporter cassette here. We're actually making cuts in the cell's genome, and you do that right before the GAPDH open reading frame and right before the CD4 open reading frame. These two genes sit about 1/4 megabase apart from each other, and when those two resulting breaks are repaired, if non-homologous end joining repairs them, it can create a gene conversion event where your GAPDH promoter, which is constitutively active, is driving expression of CD4 in cells. This system is amazingly clean because the cell lines we use aren't related to the immune system at all, and so they have no CD4 expression until we put both of our Cas endonucleases in. And then we see a very clear CD4 signal. I felt really good about myself. We were about to find out that I was right. Unfortunately, Changquen and Taylor found exactly the opposite. So it turns out that beta E6 actually blocks this pathway too. You can see our vector control cells here, lots of CD4 expression by that dark band. 86, virtually a white slate. When we correct for transfection efficiency, it gets a little better, but still 86 beta HPV is blocking about 75% of your cell's potential to use the backup system to repair double-stranded breaks. Chunquin and Taylor weren't satisfied in just proving that it was wrong. They wanted to know all the details because they're good scientists. And so they dug into this further and found out how beta HPV was working. It reduces the activation of this essential non-homologous end-joining protein, DNA PK. You can see that here, where again, phosphorylation of this protein is going to indicate that it's activated. And if you compare to the vector control cells on the left side, after double-stranded breaks, 86 is notably decreasing the amount of DNA PK activation in the cells. This doesn't actually stop the cells from forming non-homologous end-joining complexes. They do get repair complexes where DNA PK forms at sites of double-stranded breaks. Here again, I'm marking double-stranded breaks in green. Our non-homologous end-joining factors are in red. And just like happens in treated vector control cells, beta HPV expressing cells have no problem recognizing the damage and forming repair complexes. The trouble came in resolving these complexes, and Chunquin and Taylor showed that there was a notable decrease in the ability to resolve these complexes. Just had somebody tune in from Germany. That is super cool. Welcome, Baki. Okay, so summary at this point in the talk is basically that beta HPV6 has a pervasive ability to block the cell's ability to respond to DNA damage. And if you think about that in kind of the global sense, this makes a lot of sense. So you've got a virus that's infecting skin cells that needs those skin cells to grow, and it's essentially doing everything it can to avoid the cells pausing the cell cycle. And it's doing this by blocking their repair response. And you know, if it weren't 2020, that might be the end of the story, but it is 2020, and there's always more bad news to come. Not bad news in this case, really, it's just more work from a really talented graduate student in the lab, Dalton Dacus, who wanted to know what else the loss of P300 did. Immune, blah, 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 repair, blah, blah, blah. What Dalton wanted to know was how P300 affected the expression of proliferative genes. Here's another one of those volcano plots here, a little bit simplified. Dalton pulled out genes involved in proliferation, color-coded them in these blue dots, and you can see a prevalence of these genes over here in the high expressed significant range when P300 was reduced in cells. Dalton confirmed this in a cell line that was wild type for P300 or had P300 knocked out. And indeed, the expression of pro-proliferative genes increases when you remove P300 from the equation. So P300 negatively regulates proliferative gene expression. Now, these genes all had something in common. They were controlled by the hippotumor suppressor pathway, which is a master regulator of proliferation in your cells. Essentially, when your body gets any stimulus, cells in your body get any stimulus that tell it not to grow, they activate this pathway, and the hippo pathway turns off pro-proliferative genes. One of the ways that the pathway can be activated is if cells fail to divide upon entering mitosis. Now, here's a picture or a short video of normal mitosis. It's from work not related to the beta story, but I just think it's so cool that I wanted to show it here. You can see those centrosomes sitting on the outside of the bulk of the DNA in blue, pulling the DNA into two different daughter cells. Now, about 5 to 10% of the time, your skin cells, at least in tissue culture, will fail to undergo that division, that cytokinesis to become two cells, and you'll get a cell with two nuclei. This isn't so good, so your cell activates that hippotumor suppressor pathway to restrict growth and even to promote apoptosis. Specifically, it activates this LATS kinase. You can see that here with phosphorylated LATS being low before failed cytokinesis, but during failed cytokinesis, the levels of phospholates have risen quite substantially. 86 attenuates that. Again, this is the story with 86, with beta HPVs. It's not that they completely knock out a pathway, it's that they make it work much less efficiently. The ultimate result of this is that beta HPV6 limits the stabilization of p53 in response to failed cytokinesis. I'm showing some data for that here, where you can see a buildup of p53 in our vector control cells during failed cytokinesis that is absent in the cells expressing 86. Now this is important because p53 stabilization is what triggers apoptosis in response to that failed cytokinesis. And if you look 6 days, which is what the original study did, these early time points, 86 is really good at promoting the survival of cells that have failed cytokinesis. But what Don and Elizabeth and the lab followed this work up by showing was that if you're just a little more patient and follow it out to 10 days, there's really no difference. 86 is only providing this short-term protection. And when we took things out to about a week and a half, all of the cells that we induced failed cytokinesis in failed to continue growing. So they started looking for ways that they might survive. And they did this using a computational approach that showed them that telomerase activation occurred often in cells, and they thought this was a good target or potential for helping cells survive failed cytokinesis because TERT activation promotes proliferation and it helps cells avoid replicative senescence. And indeed, when they combined the two, when 86 was combined with TERT activation, the cells were able to survive failed cytokinesis 9 for 9. We did this 9 times because the assay goes out months of keeping these cells growing. Only 1 time did TERT activation on its own keep the cells growing. So this really is a synergistic relationship that can exist if you were to get a beta HPV infection in a cell that had a common mutation that exists in skin cells. That's pretty cool, but it's not so good for your cells. It wasn't just a tissue culture trick. The cells that failed to divide experience a lot of aneuploidy and a lot of micronucleation. You can see that 86 on its own actually induces a large amount of aneuploidy, shown here in red. And if you combine that with failed cytokinesis, 100% of our cells had aneuploidy genomes. This also results in micronucleation. Micronuclei, you can see here, is these little pieces of DNA excluded from the primary nucleus that are a classic hallmark of destabilized genomes. Now, the ultimate summary of this talk is that life in the fast lane comes at a price, essentially by promoting continual proliferation in the face of signals telling yourself to do otherwise. Beta HPV causes a lot of damage. Moving forward, we're interested in characterizing that mutagenic consequences in more detail, in part by some next-gen sequencing projects that we have active. We have created a double-stranded break at a defined locus, and then are sequencing the half megabase surrounding that to define how the repair is occurring there. And then we're also going to use it to define how the resolution of micronuclei occurs, because this is suspected to induce chrombotripsis, which results in thousands of clustered DNA mutations. So that's really the end of my talk. I wanted to thank you for laughing at my corny jokes, although I don't honestly know if anybody laughed because this is Zoom and you were all polite and muted yourself. But I'm just going to tell myself that you laughed, because otherwise I'm that sad man telling jokes alone in an office building. I do want to take some time to seriously thank my mentors throughout my time as a scientist. When I entered Prescott's lab as a rotating graduate student, I literally had never seen a pipette before. With his tutelage, I was able to get several papers out and convince Denise Galloway to take me on as a postdoc at the Fred Hutch, where along with Ann Roman, she mentored me to the point that K-State was interested in giving me a chance to start my own group here, and provided me Raleigh Clem as a faculty mentor. Raleigh's been wonderfully helpful in helping me navigate the craziness that comes with being an assistant professor. And finally, Geelong isn't an official mentor, but the man has read literally every grant I've ever written. And if you do that, you deserve some public praise. Thank you, Geelong. You are a wonderful colleague. And I have to end by saying thanks to my family. I absolutely love you, Chloe and Edison. You're my world. And with that, I just want to thank everybody and open it up for questions. Hopefully I didn't go too long. Let me know what I can tell you more about.
Thank you, Nick. I'll unmute everybody so we can give him a hand.
Thanks, everybody.
That deserves muting everybody again. Any questions for Nick? You're welcome to unmute yourself and ask him a question since we're running out of time here.
I have a question. This is Richard Wong at UT Southwestern in Dallas. Can you hear me?
Yes.
So my excellent talk, Nick. Do you have any information, or has anyone done studies on whether these mechanisms in terms of inhibiting DNA damage response and repair might be conserved in the gamma, mu, and nu kind of cousins of beta HPV?
Richard, that is a wonderful question. Unfortunately, to my knowledge, the answer is not really. I don't think there's been a lot of mechanistic studies, especially in mu and nu. There may have been some new stuff that I've missed in gamma. Anybody in the chat that happens to know that, feel free to chime in and fill in my ignorance. But I'm sorry, Richard, I think that the answer is that we're not certain about how well this is conserved across the different genera. But I can tell you that among species within the beta papillomavirus genus, some of these phenotypes are and some of them are not conserved.
Beth has a comment here. I don't know if it's a comment or a question, but still want to hear about pathway switching.
Absolutely, thanks, Beth. So here's a cool thing that I didn't talk about because getting into the details of this was gonna let me not talk about Dalton's work. So what Beth is referring to is the fact that we've observed that non-homologous end-joining and homologous recombination both don't work. So we have preliminary data that shows what beta HPV actually does is to cause cells to try and use both pathways. We think that non-homologous end-join is starting in cells and it gets to that DNA PK step and then fails. And we have some evidence that homologous recombination proteins are actually being recruited to the sites of damage. We can see progression through multiple steps in the homologous recombination pathway. And I'm a super geek, so that gets me excited. The reason it should matter to anyone here is that those pathways work in absolutely competing fashion. So non-homologous end-joining gets rid of all overhangs. Homologous recombination creates them. And so if they're both happening at the same site of damage, what we think is happening is we're going to get large stretches of deletions as they kind of chew up and create overhangs. That's actually the purpose of the next-gen sequencing that we're doing. So that's the like 5-second answer, but it's really cool.
So any other questions? One more question? If not, thank you very much, Nick, and we'll see everybody, I guess, next week.
And thank you all. Thank you so much.